morning everyone. Today we are going to see one topic of chapter sexual reproduction in flowering plants, and that topic is very important, double fertilization. Flowering plants are angiosperms. These angiospermic plants have a unique phenomena of fertilization that is known as double fertilization. It is not observed in any other group of plants. So what is the double fertilization? It involves two fusions, fertilization and triple fusion. Fertilization and triple fusion, these are the two fusions which take place in the embryo sac of a ovule in the pistil, that is the female part of a flower, and that is called double fertilization. So we know that pollination is the transfer of pollen grains, transfer of pollen on stigma. So when stigma becomes receptive, at the same time when pollen is released from the anther, then this process takes place. So in this process we will see the diagram that pistil which contains, I am showing it with single ovule in the center, bitegmic, and it consists of a central body, oval body, that is known as embryo sac. This embryo sac consists of 8 cells, right? And then pollen grains fall here on the stigma. These pollen grains, when fall on the stigma first of all recognizes the pollen grain if it is of right type. Right type means if the pollen grain is compatible or if the pollen grain is of the same species. Then it will be followed by the germination of pollen grain on the pollen on the stigma, and it will form pollen tube. There are many pollen grains which grow or germinate on the stigma, but out of these only one is able to reach to the ovule. Now you can see there is one opening. There is one opening, this part where these two integuments of the ovule are absent, and this opening is known as micropyle. Pollen tube is carrying three nuclei. The first nucleus is called tube nucleus, and it is followed by two nuclei which are known as two male gametes. When pollen grains germinate on the stigma, the entire content, cytoplasm, tube nucleus and two male gametes, they are transferred into the pollen tube, and this is growing towards the ovule through style. So this style is made up of cellular part, and this through the cellular parts pollen tube grows downwards towards the ovule. So what is this structure? Ovule. What is this basal part? That is ovary. So stigma, style, ovary and ovule, and inside the ovule we have a central oval body which is known as embryo sac. This embryo sac is actually the site of double fertilization. It is actually the site of double fertilization. So now let's see what are the cells present inside the embryo sac. These are cells which are called antipodals. These are 3 in number. These two central cells are polar cells. They are 2 in number. Here the central cell is known as egg or the female gamete, which is 1. And then we have 2 cells here below the egg synergids. These are also 2 in number. All these cells are haploid. Each polar cell is haploid, egg cell is haploid, synergids are also haploid, right? So this is 8-celled embryo sac which consists of these cells, 3 antipodals, 2 polar cells, 1 egg cell and 2 synergids. This is the situation when the pollen tube is growing downwards and moving towards the ovule and takes entry through the micropyle. Now this part we will see here, like in the embryo sac when we have 2 synergids, these synergids contain an apparatus which is known as filiform apparatus, and here we have egg. So this apparatus is filiform apparatus. It has very important role in fertilization. Right? These, when pollen tube touches the wall of the embryo sac, these filiform apparatus, they secrete chemical which dissolves this wall. So pollen tube is taking entry from here and it is having 2 male gametes. So these filiform apparatus, they secrete chemical which will dissolve this wall. So we can see a small change here. This wall will get dissolved and it will make a pathway for the entry of both the male gametes. At this time there are 3 simultaneous events taking place. One, this is dissolving the wall of the embryo sac, and that is followed by the degeneration of both the synergids. Both the synergids disappear from here. What will remain behind? This is the egg cell. And at the same time, both the polar cells will fuse and they will form single polar cell. So this polar cell is the result of 2 polar cells, so it becomes 2N. That means it is formed by the fusion of two cells. So it is now diploid, but egg will remain haploid. Right? At the same time, these 3 antipodals also degenerate. So we have to see 3 things which are taking place simultaneously. Number 1, synergids secrete chemical to dissolve the wall of the embryo sac and they degenerate. At the same time, both these polar cells fuse and form a single polar cell, that is going to be a diploid cell. Antipodals also degenerate. So we have this embryo sac at this position here. So you can see there are only 2 cells left in it. Now we can see the movement of these 2 male gametes which are present here. They will be transferred, these 2 male gametes will be transferred here in the embryo sac. Fine. So in next situation you will see that this embryo sac has 1 egg cell, 1 polar cell, and there are 2 male gametes. One comes and fuses with the egg cell, the other one, second male gamete, fuses with the polar cell. So what is our labeling now? This is showing polar cell and this is male gamete. They are fusion, and here it is showing the fusion of 1 male gamete and this is the egg. Fine. So this fusion is called syngamy or fertilization, and this fusion, because it is involving the fusion of 3 cells here, 2 polar cells, 1 male gamete, 3 cells fusion, triple fusion, fusion of egg cell and 1 male gamete, fusion of 2 cells, that is syngamy or fertilization. Because there are 2 fusions and both of these fusions are collectively called double fertilization. Right? So in the next figure we can write it this way: when egg fuses with male gamete, it will form zygote. And second phenomena is polar cell fuses with other male gamete and it gives rise to primary endosperm nucleus, which will finally grow in a triploid tissue, endosperm. So what is the change in the ploidy of these cells? This is haploid, and zygote is going to be diploid. Now these polar cells, this is diploid, this is haploid. So 2 plus 1, 3, and this tissue is going to be triploid tissue. So endosperm is also triploid tissue. So what is this phenomena called? It is called fertilization. And this one is called triple fusion. And both of these are collectively called double fertilization. So fertilization plus triple fusion are known as double fertilization. What is the significance of this phenomena? We can easily see here that when zygote formation takes place, simultaneously endosperm formation takes place, right? Endosperm is a nutritive tissue. It is a nutritive tissue. So it is formed simultaneously because when zygote will grow into embryo, this endosperm will provide nutrition for the development of embryo. So there will be two ways, either it is endosperm is consumed at the time of development of embryo or it is consumed at the time of germination of the embryo. So that differentiates between endospermic and non-endospermic seeds. But what is the aim of double fertilization? It is just like nature has made the arrangement of food for the zygote when it is developing into embryo. So this entire event is double fertilization. We see once again this recap of this entire topic. Pollen grains fall on the stigma. They germinate because the stigma is receptive. It is recognizing its pollen grain, allows to grow pollen tube which comes down through the style, enters the ovule through micropyle. Synergids secrete from philia apparatus chemical that dissolves the wall of the embryo sac. Synergids degenerate. Two polar cells fuse and form one polar cell, diploid polar cell. Anticotyls also degenerate. And now these male gametes, they find their way into the embryo sac. One moves towards the polar cell, the other one moves towards the egg cell. Fusion of egg cell and male gamete, that is fertilization or syngamy. Fusion of male gamete and polar cell, that is the triple fusion. So there are two fusions in the embryo sac. Two fusions in the embryo sac, fertilization and triple fusion, and that is why it is called double fertilization. Do practice of this diagram at least twice or thrice in your register. And this is the diagram which is asked by a question. Draw the diagram of pistil showing the events of double fertilization. Thank you.